So, what do you think powers every single move you make? I mean everything, from blinking your eyes to running a marathon. Now, the obvious answer might pop into your head, but the real story, well, it's way more complex, and it's hidden deep inside our cells.
It's not just about the protein that builds your muscles or the fat you burn as fuel. Sure, those are a big part of the picture, but they're standing on the shoulders of a much more fundamental and honestly often overlooked biological architecture.
The real secret, the powerhouse behind it all, is a hidden world of complex sugars. And no, I'm not talking about what's in your soda. We are talking about incredibly sophisticated sugar blueprints that literally build our bodies and power our lives.
And that is exactly what we are diving into today. Polysaccharides. Think of them as the body's essential sugar blueprints.
So here's our game plan. We'll start with the sweet basics of carbohydrates, then look at the simple sugar units that act as building blocks. We'll see how they form these massive chains, explore what they do beyond just giving us energy, and finally look at what happens when these intricate biological pathways go wrong.
All right, let's start at the very beginning. Before we can get into the really complex stuff, we have to ask a pretty simple question. What is a carbohydrate on a chemical level?
At its heart, it's a molecule made of just three things: carbon, hydrogen, and oxygen. And that general formula, CmH2ON, kind of gives away the name. Carbo for carbon, and hydrate for water. This simple recipe is the foundation for an absolutely incredible amount of diversity.
You know, a really key difference between sugars comes down to where a specific double-bonded oxygen atom is placed. If it's at the end of the carbon chain, we get an aldehyde, like glucose. If it's stuck in the middle, we get a ketone, like fructose. This tiny shift completely changes the molecule's chemical personality, which is absolutely crucial for how our body can use it.
Okay, now here's a concept that might bend your brain a little. Handedness. Just look at your hands. They're perfect mirror images of each other, right? But you can't lay one flat on top of the other and have them match up perfectly. Well, some carbon atoms in molecules have this exact same property, creating a left-handed and a right-handed version of the same molecule.
And here is where it gets absolutely amazing. Life chose a side. The amino acids that build every protein in your body are almost all left-handed. But the carbohydrates that fuel our world, they are almost universally right-handed. Why? Honestly, we don't fully know, but it's one of the fundamental rules written into the code of life itself.
So, with that in mind, let's zoom in and look at the individual bricks that build this entire sugar world, the simple sugar units, which we call monosaccharides.
Now get this. All the sugars we're about to look at share the exact same chemical formula, C6H12O6, 6 carbons, 12 hydrogens, 6 oxygens. And yet, they are completely different molecules. So how is that possible? Well, it all comes down to tiny but critical differences in their 3D shape. They're what we call isomers. Let's put two of them side by side. Glucose, our body's main fuel source, and galactose, the sugar you find in milk. Look closely. The only difference between them is the direction of this one little OH group on the fourth carbon. In glucose, it points to the right. In galactose, it points to the left. That is literally it. And here is the punchline. This is so important. That single tiny flip means that the enzymes in your body that are perfectly shaped to break down glucose are completely blind to galactose. It's like having a key that's just one tiny groove off. It simply will not fit in the lock.
And this isn't just a fun fact for chemistry class. It has life-or-death consequences. In the genetic disorder galactosemia, the special enzymes needed to process galactose are broken. This causes galactose to build up to toxic levels, leading to severe damage. It's exactly why we screen newborns for this condition.
So these long, straight chains of sugar, they're actually not very stable in the watery environment inside our cells, so they do something really clever. They basically bite their own tail. The top of the molecule reacts with the bottom, and it curls up to form a much more stable ring.
But wait, there's one more twist. When that ring closes, the new OH group that's formed can either point down, which we call the alpha isomer, or it can point up, which we call the beta isomer. Now, please remember this alpha versus beta distinction. I promise you it is about to become incredibly important.
All right, so we have our individual building blocks, our little sugar rings. Now let's see how they connect together to form the massive structures we call polysaccharides. The glue that holds these sugar units together is a special connection called a glycosidic bond. It's formed when two sugars link up, and in the process, they release one little molecule of water. This is how we build everything from simple two sugar pairs to gigantic polymers.
And here's where sugars get way more complex than something like proteins. With proteins, the bonds are pretty standard, but with sugars, the details of the bond matter immensely. You have to say which carbons are connected and whether it's an alpha or a beta connection. This ability to link up in so many different ways is what gives them such incredible structural diversity.
We basically classify these chains based on their size. If it's just two sugars linked together, that's a disaccharide, like table sugar. A short chain of a few sugars is an oligosaccharide. And the really huge, long-chain molecules, those are the polysaccharides.
And this slide right here perfectly illustrates why that whole alpha versus beta thing I told you to remember is such a huge deal. Just look at glycogen and cellulose. They are both made of the exact same monomer, glucose. But check out the bond type. Glycogen uses alpha bonds, which creates a branched, sprawling structure that's perfect for storing energy. Cellulose uses beta bonds, which creates these rigid, straight fibers that give plants their structure. Your body has enzymes to break alpha bonds, but not beta bonds. And that one simple difference is why you can digest a potato, but you can't digest wood. But sugars aren't just for storing energy or building the walls of plants. They also form the sophisticated, gel-like scaffolding that literally holds all of our tissues together.
I want you to meet the glycosaminoglycans, or GAGs for short. These are long, repeating sugar chains that fill the space between our cells. Now, their secret superpower is that they are packed with negative charges. These charges repel each other and suck in a ton of water, creating this amazing, springy, shock-absorbing gel that cushions and supports all our tissues.
And trust me, you've encountered these gags your whole life without knowing their name. Hyaluronic acid, that's the lubricant in your joints and the goo that fills your eyes. Chondroitin sulfate gives your cartilage and tendons their incredible strength. And keratan sulfate is a key player in your cornea, bones, and even your nails.
But the complexity doesn't even stop there. When you take these long GAG chains and attach them to a central protein core, you form an even bigger, more complex molecule called a proteoglycan. They look like giant bottle brushes, and they are absolutely essential for the structure and function of pretty much every tissue in your body.
Okay, so it's clear these sugar molecules are critical for energy, structure, and so much more. But what happens when the complex factory inside our cells that builds and recycles these sugars breaks down?
Well, that brings us to a really tough family of genetic disorders known as mucopolysaccharidoses, or MPS. In these diseases, those GAGs we just talked about can't be broken down and recycled properly, so they start to accumulate inside the cell's recycling center, the lysosome. And this buildup becomes toxic. And the consequences of this molecular traffic jam are severe, and they affect the entire body. Patients often suffer from developmental delays, serious skeletal problems, enlarged organs, and have very distinct facial features, all of which usually start to appear within the first few years of life.
Now, here's how it's supposed to work. It's like a perfectly efficient disassembly line. A GAG goes into the lysosome, and then a whole series of specialized enzymes goes to work, each one snipping off a specific piece of the chain, one after another, until the whole thing is broken down into parts the cell can reuse.
But in MPS, a single genetic typo means that one of those enzymes on the line is either broken or missing entirely. This grinds the whole disassembly line to a halt. The process stops cold, and these partially degraded GAGs just start to pile up and up, eventually causing the cell to malfunction and die.
So how do doctors figure out which enzyme is the problem? Well, they can start with a simple urine test to see if there are high levels of GAGs. Then they can do a more specific enzyme activity test on a blood sample using fluorescent markers to see exactly which enzyme isn't working. And finally, genetic testing can nail down the exact mutation in the gene, giving a definitive diagnosis.
And the reason getting that diagnosis early is so incredibly critical is because this isn't a hopeless story anymore. While these diseases are devastating, medical science has developed a pretty incredible arsenal of therapies to fight back.
I mean, these therapies are really at the cutting edge of what's possible in medicine. We're talking about things like stem cell transplantation, enzyme replacement therapy where you just supply the missing enzyme, and even gene therapy to try and fix the original genetic mistake. And then there's a particularly clever approach called pharmacological chaperones.
Let's just pause on that last one because it's so cool. See, sometimes the genetic mutation doesn't completely destroy the enzyme. It just causes it to misfold, kind of like a crumpled up piece of paper. A pharmacological chaperone is a small molecule that can bind to that faulty enzyme and act like a molecular splint, helping it fold back into a more correct, more functional shape and restoring at least some of its activity.
It's just a beautiful, elegant solution to a really complex problem.
So we've gone on this incredible journey, all the way from the simple flip of an OH group on a sugar molecule to the frontiers of gene therapy and molecular chaperones. It really makes you wonder, doesn't it? What other hidden biological blueprints are all around us, just waiting for us to finally learn how to read their code?